Hello, my name is Hratch Baghdassarian. I'm a postdoctoral fellow in the Laufenberger Lab at MIT. And today I'm going to be talking on my work on some algorithmic developments to model signaling pathway activity at single cell resolution. And to motivate this, it's important to note that signaling pathway activity is highly combinatorial at genome scale. So on the left here, we're seeing an image where multiple ligand stimulants are interacting with multiple receptors simultaneously. And on the right, we see downstream of that intracellular pathways can further experience crosstalk. And so for multicellular organisms, these many-to-many interactions help to yield specificity in a response, and they also help provide a large repertoire of functional outputs. However, you can imagine that given the large number of possible interactions, this becomes intractable experimentally to decipher the activity. Furthermore, these many-to-many interactions can enable context specificity. And what I mean by that is that given a particular cell type or disease state, the same stimulant can induce different downstream responses. And this is due to varying expression of pathway subcomponents. And so in summary, we have this large combinatorial space of relevant interactions, as well as context specificity, all of which make this very amenable to a computational approach. However, you can imagine with modeling, there tends to be this trade-off we can achieve between mechanistic detail and the breadth of various signaling pathways we can capture. So on the right here, we're seeing an example where perhaps we're capturing more pathways, but missing some of the details in those pathways. And so a former postdoctoral associate in the Laufenberger lab, Avlant Nilsson, developed an approach to address this trade-off using neural networks. And the key is that the neural network architecture is built on a prior knowledge protein-protein interaction network. And so this sort of becomes a gray box neural network model that achieves both mechanistic detail and good predictive accuracy. And so specifically, this model takes as input ligand concentrations or really any sort of signaling pathway perturbation and provides as output predicted transcription factor activity. And as I mentioned, this is now genome scale and mechanistic. And so the goal of the work I've been doing is to firstly help generalize this across multiple contexts, so different cell types, et cetera, because currently a different model needs to be trained for example, for any given cell line. And secondly, if we're thinking about single cell resolution to help capture the heterogeneity in these single cells. And so the way that we're doing this is that first we take some sort of prior knowledge protein-protein interaction network. In our case, we've been using OmniPath. Now these tend to have known directions of the weights. So we know whether an interaction is activating or inhibiting at times. However, they're not quantitative, so we don't know what the strength of those interactions are. So we can take this prior knowledge network, represent it mathematically as an adjacency matrix, and then the weights between these protein nodes become parameters that the model learns to assign a quantitative value to. This is then combined, obviously, with our signaling perturbation, and these two together account for stimulation information. And finally, we add in a bias term here. And so we can think of the adjacency matrix as the sort of strength of an interaction if it's occurring. And so this is the more context-independent term, whereas the bias is the amount of any given component needed for the interaction to occur. So this is more of that context-dependent term. These three terms are combined and run through a nonlinear activation function, which is common practice in neural networks and also relevant to signaling interactions. And we specifically use a Michaelis-Menten-like activation function in order to make this a little bit more biologically relevant. And finally, all of this is run through a recurrent neural network. And the idea here is to capture possible feedback loops in our signaling pathways. And then the model is going to essentially minimize some sort of a loss term, which defines a discrepancy between the model prediction and the actual values, and the parameters are going to become learned in a manner that minimizes that loss. And now to capture that heterogeneity I was talking about, we employ a variational autoencoder. So this takes as input our gene expression vector and provides as output learn parameters for a given distribution. So for example, for a normal distribution, we'll have our mean and our variance. And then from this, we can generate our bias term. Furthermore, to capture that context specificity, what we want to do is actually remove first the categorical information from that bias term. So we call this a global bias term. And because you can imagine that the gene expression input has a lot of categorical information in it, and then we'll add this back in using learned categorical embeddings. And I'll talk a little bit more about how that removal happens in a few slides. But with this setup, we can ask this question of, given a cell's gene expression under one particular stimulation condition or cell type, what would it look like under a different stimulation condition or cell type? And so to assess the model, we've started off with a somewhat simple single-cell RNA sequencing data set. It's eight pooled lupus samples, which had their PBMCs measured, and we can see that there's a number of different immune cell populations represented here. And then these samples were either unstimulated or stimulated with interferon beta, and we can see that that stimulation provided very strong separation in UMAP space. And so I'm going to spend the rest of this presentation first talking about how this adversarial training looked like, and then what our actual predictions look like. So for adversarial training, the goal, as I mentioned, is to remove any sort of context-specific information from our generated global bias. And so in our case, that's removing cell type. And the way that this can be done is that we can train another neural network, a discriminator, to classify the generated output as a given cell type. And the goal of the encoder or generator is to create a global bias term that is good at tricking this discriminator, which results in a vector that's devoid of categorical information. Mathematically, the way that this is done is that the generator is trying to maximize the loss of the classifier. And we can see across training epochs that the curve of the discriminator starts off at random, the loss curve, and then it goes down to near zero, meaning that it's learning how to classify this generated output. And then as the generator gets better at tricking the classifier, it returns back to approximately random, represented by this gray dashed line. And so then we can take that discriminator loss and, as I mentioned, use it as a penalty to maximize the loss with the generator. And we can see in the actual reconstruction curve or the total loss curve that the difference in the total loss without adversarial training and that the final total loss is that adversarial penalty. Now we can also assess this visually by taking that global bias term and observing what it looks like in UMAP space. So if we take that global bias term, we can see that across the various cell types in the test data, in this example, the cell types are very well mixed. Whereas when we add that categorical bias back in, we achieve separation by cell type. And so this is telling us that the global bias is devoid of categorical information and that the categorical embeddings are learning the relevant cell type information. Now, going into the actual predictions, I found that actually just observing these things visually in PC space is often a really good assessment. And so I'll show a leave one out example with dendritic cells, where we're trying to predict unstimulated dendritic cells from stimulated dendritic cells. So in this PC plot, we're seeing all of the training data, and highlighted in orange is the actual stimulated dendritic cells from which we're generating our predictions. And then in green is our actual test data of our unstimulated dendritic cells that we're going to compare our predictions to. And keep in mind, because this is generative, we don't expect a one-to-one mapping, but to actually just match the population distribution. And then finally, in red, I'm showing a projection of our predictions. And what we can see is that we are achieving both cell type and perturbation specificity, and capturing that variance at least in that PC2 direction. This can be assessed quantitatively in a number of different ways, one of which is to compare to baseline models. And so here I've compared the prediction loss to a permuted output, a linear model, and a feed-forward neural network. While I haven't shown the statistics here, the single-cell LEMBA's full prediction is significantly has a significantly lower loss than the other three models, indicating that it's performing better. Now, the final thing I want to spend a minute or two talking about is actually assessing how each component of our model is contributing to that prediction, because we have certain expectations of each of these components. So for the adjacency matrix, we want that to capture our simulation information. The global bias, we want to capture our variance, um, and the categorical bias, we want to capture our cell type information. However, you can imagine scenarios where some of these learned parameters outweigh the rest and are just capturing all of the information together, which we don't want. So first, we can assess this in general by seeing whether removing each component makes our predictions worse. And that's what I'm showing here, where we have our loss on the y-axis and then each removed component on the x-axis. The first is the full model prediction, which is also highlighted by a red dashed line. And what we can see is that removing any given component makes our loss worse, which means that each component is positively contributing to our prediction. Next, just visually, we can look at those projections in PC space to see how each component is contributing to the model. So for example, if we remove our categorical bias, what we can see is that we still have our variance and some stimulation. However, we've shifted out of the localization of the dendritic cells. And then if we retain our adjacency matrix and our categorical bias, but we remove our global bias, we're now at that centroid of our test data. However, we no longer have any sort of variance because we don't have the global bias. And finally, if we have just the adjacency matrix, we're in that plane of stimulation, but we don't have variance or cell type specificity. So these are all indicating to us that we're actually capturing the relevant information with each component of our model. Finally, I just want to acknowledge a number of my colleagues in the Laufenberger lab, in particular Nikos, who recently received his PhD in biological engineering and has provided a number of useful insights for me, as well as Avant, who I mentioned earlier, who's now a professor at the Karolinska, and has also provided a lot of useful input for me in developing this.